Bismillahirrahmanirrahim. Regulation of gene expression in prokaryotes. It is continuation of the previous lecture. Here we discuss about the other transcriptional process which are involved for the regulation in prokaryotes. This is the summary of the previous lecture, that how lac operon it is regulated in the presence of both glucose and lactose, in the presence of glucose and in the absence of lactose, even in the absence of both glucose and lactose, in the absence of glucose and presence of lactose, how different situations impact on the expression of this lac operon, and also this lac operon it is regulated. Now next is the regulation through the formation of attenuator. This structure is also known as the attenuator stem and loop structure, hairpin structure, and these structures also involve row independent termination of the transcription of almost all the prokaryotes. But this structure is also involved for the regulation of tryptophan operon. The tryptophan operon consists of genes of 5 enzymes. So 5 coding segments, they are present in the tryptophan operon along with the regulatory sequence or the leader sequence. Upstream of these 5 genes, there is a sequence which is known as the regulatory sequence, and this regulatory sequence it is uh, uh, present upstream, and when this is transcribed and translated, then here is the regulation is present, how tryptophan operon will be worked. It is another type of the negative regulation. These enzymes which are produced, the gene product of these enzymes, these are involved for the synthesis of tryptophan. This attenuator, when formed between the segment 3 and 4, it is effective to stop the translational process. So there is no translation of the proteins for the formation of tryptophan amino acid, because already tryptophan is present in the medium at high concentration. But if there is a low concentration of tryptophan, then what happens? How these genes, they are regulated through this attenuator. 2 codons present in the at the segment number 1 of this leader sequence of messenger RNA for tryptophan operon. When there is a low concentration of tryptophan, then the ribosomes are difficult to find this tryptophan from the medium, and so they paused at segment number 1 for a while, and it gave the time for segment number 2 and 3 to form this stem and loop or structure or attenuator. This attenuator structure now formed between segment number 2 nucleotide sequence and segment number 3 nucleotide sequence instead of 3 and 4 segment numbers. So these the formation of these attenuator, it is not so much effective so as to halt, to inhibit this translational process. And if this attenuator is formed between segment number 2 and segment number 3, then the translational process it is going on, and these genes, they are further translated for the messenger RNAs continue, and these genes, they are further translated to form a polypeptide chain, so as these polypeptides are actually the enzymes for the synthesis of tryptophan. This happen at low concentration of tryptophan, but this happen at high concentration of tryptophan. At high concentration of tryptophan, segment number 3 and 4, they form the attenuator, while at low concentration of tryptophan, segment number 2 and 3, they form the attenuator and continue the transcription as well as the translational process. This is one type of negative regulation in case of prokaryotes like bacterium, and this regulation is according to the situation and circumstances of the medium. Here another way to show that how this messenger RNA is formed, how this attenuation is formed between 3 and 4, and how the stop or termination of RNA polymerase so as to stop the messenger RNA. When there is no messenger RNA, then no polypeptide chain has been formed. The transcriptional and translational process, they are going side by side in prokaryotes. As the messenger RNA is formed or transcription is going on, the messenger RNA that has been formed and undergoes the translational process. So these two processes, they are going side by side. Another regulation in prokaryotes is the stringent regulation or stringent response. The prokaryotes, their responses according to the situation of the medium, according to their environment, according to their circumstances. Sigma factors in prokaryotes, they recognize different promoter region. So according to the situation, sigma switching take place. When there is a deficiency of when there is a deficiency of amino acids, and these amino acids, because they are deficient, so there is a different types of regulation or gene expression take place. The genes that are the A site of the ribosomes, they bind with the uncharged tRNA. When there is a deficiency of amino acid in the environment, then the A side, it binds the uncharged tRNA, and this uncharged tRNA when bind, then the series of reactions take place which activate the rel-A protein. Rel-A protein, basically it is the enzyme or it is the protein, catalytic protein that bind physically with the ribosomes. When binding to uncharged tRNA during the deficiency of or scarcity of amino acid in the medium, it activates the rel-A protein. This rel-A protein, basically it is the stringent factor, and it converts the guanosine diphosphate into the guanosine tetraphosphate. This is alarmone guanosine tetraphosphate. This alarmone guanosine tetraphosphate undergoes the inhibition of ribosomal RNA and tRNA and some of the messenger RNA, so as no proteins are formed for these in during these situations. So one of the stringent response is the production of rel-A protein so as to form the alarmone guanosine tetraphosphate, and this alarmone guanosine tetraphosphate, it is used for the inhibition of ribosomal RNA and no tRNA, some of the messenger RNA, so there is no translational process take place. And other response is the when there is a balance, transcription and translation coordination. Transcription and translation coordination is that when there is a balance between the ribosomal RNA and the ribosomal proteins. The proteins, they are involved for the association of ribosomal RNA so as to form the ribosomes, and these ribosomes are now involved for they act as a translational machinery for the synthesis of protein. When there is a high concentration of ribosomal protein, these are the operons for ribosomal proteins, and these ribosomal proteins also known as the R proteins. When there is a high concentration of ribosomal protein, production of high ribosomal proteins, then what happen? They themselves negatively control their translation. How? One of the ribosomal protein or R protein, they join the Shine-Dalton sequence of this Shine-Dalton sequence present upstream of the messenger RNA in prokaryotes from the initiation codon or the start codon. When they bind with the Shine-Dalton sequence, then they inhibit this messenger RNA so as to translate it. And along with, there is also the assembly of the small units of these ribosomes with on these Shine-Dalton sequence. How so these messenger RNA, it is inhibited through translation by the binding of these proteins R proteins on these messenger RNA. Another process is that they also inhibit their own messenger RNA by binding these proteins with their own messenger RNA. So these messenger RNA when bind with these R proteins, when bind with their own messenger RNA, they also inhibit their translation process. By means of these different ways, the prokaryotes, they can regulate their gene expression or according to the situation, as during the favorable conditions and unfavorable conditions. If there is also another phenomena known as the sigma switching. Sigma is actually the factor which recognize the promoter region, but when there is the conditions are different, then different sigma factors, they are involved to recognize the promoter region and thus to express the different genes. Thank you very much.